So let's review genetics, the magic of the double helix, and talk about nucleic acids. So we are reminded that following the destruction of Hiroshima and Nagasaki by the US using the atomic bombs, Fat Man and Little Boy, this group of scientists then turned to the next step, which is if we can destroy all life on Earth, how do we maintain it or save it or recreate it? So the race was on for the double helix, figuring out and discovering how DNA was structured and how it functioned. So we begin with Alfred Hirschi and Martha Chase, a bacteriologist and a geneticist here in the United States, and they determined that it was DNA and not proteins that carried information. Their studies basically followed injected DNA in bacteriophages, which are viruses, and monitored how that DNA was transferred from cell to cell. That brought us to Oswald Avery, who did much of the same and figured out that it was the chromosomes and the genes that were made of or composed of the DNA. He did this by injecting different strains into mice and following that strain through the life form and seeing its effect on the mice. Brings us to Austro-Hungarian biochemist Erwin Chargaff, who was the first to realize that the numeric ratios meant that adenine and thymine and guanine and cytosine had to be complementary to each other because their numeric ratios were similar. That brings us to Maurice Wilkins and Rosalind Franklin, both working in England at the time and using a process called X-ray diffraction crystallography to take photographic images of DNA, and they were also taking pictures of RNA and not realizing it, and their discovery was key to unleashing the model and structure of DNA. And that brings us to James Watson and Francis Crick, and we discussed in class how Watson basically was good at manipulating, borrowing, and stealing information from others, and he convinced Crick to use that information to build the first model of DNA. Remember that their first model was actually quite erroneous because they did not understand the full chemistry of the model, and it was only using the picture taken by Rosalind Franklin that they were actually able to put this model together. So that image that we discussed about from Rosalind Franklin is known as Photo 51, and you should recall that we watched the documentary on Photo 51 and saw how valuable Rosalind Franklin's images provided information to Watson and Crick. We also talked about the fact that Rosalind Franklin unfortunately was not awarded the Nobel Prize, as were Watson, Crick, and Wilkins, because she passed away from cancer, most likely from the work she was doing in X-ray crystallography, and they do not award the Nobel Prize posthumously. And if you want to review or see that documentary again, here is the link. So we are reminded that inside of our nucleus we have chromosomes, 46 to be exact in the human species, and those chromosomes are made out of branch arms and legs called chromatids, and those chromatids are formed from coils and coils and coils of a model called DNA, and this DNA molecule are formed from units called nucleotides. And because of their twisted ladder structure, they are called a DNA double helix. So the actual structure of DNA, deoxyribonucleic acid, DNA, is a nucleotide, and the basic unit of this DNA, the nucleotide, is composed of three basic structures. A phosphate, which is used for bonding, a sugar which forms the backbone of the DNA model or molecule, and then the nitrogenous base, which there are 4 of, and that is actually the unit or part of the DNA that conveys the message or holds that information that we call the blueprint of life. Again, the DNA is composed of this nucleotide unit made up of a phosphate, which acts as a bonding molecule to help form the backbone of the helix. The sugar is called deoxyribose. It is a 5-carbon sugar. That's why we are representing it here with a pentagon, and that forms the actual backbone or sides of the ladder of the double helix. And then the nitrogen base forms the rungs of the ladder and spells out the genetic information for that blueprint. Now, there are 4 different nitrogenous bases, and these bases are the actual letters of the genetic message. And the 4 bases are adenine, thymine, guanine, and cytosine. Adenine and guanine are both longer because they are double rings, so they are called the purine bases. And then thymine and cytosine are actually shorter. They are composed of 1 ring, and they are called the pyrimidine bases. Now, the bases pair in a complementary form, and there are 3 reasons that they pair together in this way. Adenine only pairs to thymine. Adenine and thymine are a complementary base pair, and guanine only pairs to cytosine. Now, they pair together because of shape and size, purine to pyrimidine, and they pair together because of numeric ratio. Adenine and thymine have the same genetic ratio, as do guanine and cytosine. So adenine can only pair to thymine, guanine can only pair to cytosine. The bases are held together in the middle by hydrogen bases, and we are reminded that hydrogen bases are weak. They act almost like Velcro, which means you can pull them apart and push them back together quite easily. Adenine and thymine are held together by 2 weak hydrogen bonds. Guanine and cytosine are held together by 3 weak hydrogen bonds. Now, the ladder of DNA is composed of the sides of the ladder made up of the phosphate sugar bonds, and the rungs of the ladder are formed by the base pairs, adenine to thymine, guanine to cytosine, and it is the order of these letters that determine the proteins that you can make that build our lives. Remembering that nitrogenous bases form the rungs, complementary base pairs of adenine and thymine, guanine to cytosine, and the sides of the ladder are the phosphates and the sugars. So again, chromosomes are made out of coils and coils of this DNA molecule. They coil upon each other around molecules called histones. As they coil further, they form the chromatids that become the 4 parts of the tetrad that make up the chromosomes inside our nuclei. The second nucleic acid is ribonucleic acid, RNA. RNA has the same structure as DNA as far as its nucleotide base. It has a phosphate and sugar, but in this case the sugar is called ribose, and it also has nitrogenous bases. Now, the difference is in ribonucleic acid, we have a base called uracil, which replaces thymine in the RNA. So the complementary base pairs here will be uracil to adenine and cytosine to guanine. Uracil replaces thymine in the RNA. Now, there are actually 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA is a single strand. It actually transcribes or copies the message from DNA found in the nucleus and then carries that message out to the ribosomes, which are the sites for protein synthesis. It is this message that allows us to build the proteins that are the functional instructional units of our lives. Now, the second form is tRNA. tRNA is a single unit, and it translates the message from the mRNA into the amino acid units that become the polypeptide chains that build proteins, becoming the protein strands. So it is the combination of messenger RNA bringing the message to the ribosome and the tRNA bringing or transferring the amino acids into position that we are able to form the polypeptide chains that make the proteins that allow us to function in life. The third RNA is called ribosomal RNA, found in the ribosomes. Its job is to proofread the message as mRNA is translated to tRNA and into the amino acid chain. RNA is often called reader RNA because of this proofreading process. Finally, let us compare the two types of nucleic acids. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA has two strands, making it a double helix. RNA forms either a single strand as mRNA or a single unit as tRNA and rRNA. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. DNA uses the base thymine, while RNA uses the base uracil. DNA, there is only one type, the double helix, while RNA has three types, the single strand mRNA carrying the message, the single unit tRNA which transfers amino acids into position, and the single unit rRNA or ribosomal RNA which proofreads the message. DNA is found only in the nucleus, while RNA can be found throughout the cells.